Okay, so let's unpack this. The way a protein folds,
Right.
that intricate dance,
Well,
amino acids twisting into a specific 3D shape
it's absolutely fundamental.
to how it works, yeah.
Yeah,
Like a key fitting a lock, sort of. The structure dictates the function.
exactly.
And for decades, figuring out that fold just from the sequence, that was, well, a monumental challenge.
A huge challenge. The protein folding problem, as they called it.
Right, just this. Astronomically large number of possibilities. Made experimental work incredibly slow. Difficult.
Venki Ramakrishnan, the Nobel Laureate, he really emphasized how tough it was.
And then AlphaFold comes along.
Right, offering this computational shortcut just fundamentally changed the landscape of structural biology.
It's wild. I mean, for a protein, say, just 100 amino acids?
Yeah, relatively small.
The number of ways it could fold is, what's the number?
Order of 10 to the power of 47.
I can't even picture that number.
Nobody can. It's Levinthal's paradox, basically.
Yeah.
How does it find the right fold so quickly when there are so many wrong ways?
Before AlphaFold, anyway.
Before AlphaFold, yeah. We had to rely on these incredibly laborious techniques.
Like X-ray crystallography.
Yep, and cryo-EM, cryo-electron microscopy. Powerful but painstaking work. Mapping these structures took forever.
Exactly. And that's where our deep dive begins today, really. We're diving headfirst into AlphaFold.
The AI program that didn't just chip away at the problem?
No, it, blew it wide open.
Pretty much.
Its significance, undeniable. I mean, the 2024 Nobel Prize in Chemistry.
Awarded to Demis Hassabis and John Jumper specifically for AlphaFold. It's huge.
Think about that. An AI for protein folding gets the Nobel.
And actually, the prize recognized two complementary areas, which is interesting.
Oh, yeah.
Yeah, one was AlphaFold's amazing ability to predict the structures of natural proteins.
Okay.
And the other half went to David Baker.
For designing new proteins, right?
Exactly. Designing totally new proteins. And what's cool is that AlphaFold can kind of work in reverse, too.
Yeah.
It helps figure out the sequence you need to get a desired shape.
Wow. Prediction and design.
And we should also remember the underlying tech, the neural networks that work, also got recognized. Physics Nobel. It's all connected.
So our mission today, for you listening, is to really get what makes AlphaFold tick.
Yeah. How does it actually work?
How it works, the massive impact it's having everywhere.
The limitations, too. Got to talk about those.
Definitely. Where does it still struggle? And, crucially, what's next? What's the future hold?
We're aiming for those aha moments.
Without getting totally lost in the weeds. Ready to jump in?
Let's do it. So back to that challenge. 10 to the 47 possibilities.
It's almost incomprehensible.
It really is. And compare that to the old ways. X-ray crystallography. You need to get the protein to form crystals first.
Which isn't always easy, I hear.
Not at all. Sometimes impossible. And then you have to interpret these complex diffraction patterns.
And cryo-EM.
Powerful, especially for bigger things, large complexes, but it has its own technical hurdles. Both methods are slow, need specialist gear, expertise.
Right, and that's where CASP comes in, the critical assessment of structure prediction.
Yes, CASP. Super important.
This rigorous, independent competition.
Right.
Held every 2 years.
Exactly. A blind test. Researchers submit predictions, and they're judged against experimentally determined structures that haven't been released yet.
So no cheating.
No cheating. It's the gold standard for validation of the field. And CASP is where AlphaFold really proved itself.
So how did it evolve through CASP? Tell me about AlphaFold 1.
Okay, AlphaFold 1, that was 2018, CASP 13, made a big splash.
How did it win? What was its trick?
It focused on predicting contacts, basically which amino acid residues were likely to be close to each other in the final folded structure.
Ah, okay. Like mapping out the neighborhoods within the protein.
Precisely. It used neural networks to learn the link between the sequence and these spatial contacts. Clever approach. But the code itself, it wasn't really designed for general use outside that CASP 13 data set. More of a proof of concept, showed the potential of the deep learning direction.
Gotcha. And then came AlphaFold 2, 2020, CASP 14.
That was a revolution.
The real game changer, yeah. Accuracy comparable to experiments.
Exactly. It was a massive leap. It went beyond just contacts. Used deep neural networks to predict the entire 3D structure, end to end.
Wow.
Huge deal. Published in Nature in 2021. And critically, DeepMind open-sourced the code.
And launched the database.
And launched the AlphaFold protein structure database. That was key.
Yeah.
AlphaFold 1 showed promise with contacts, but its use was limited. AlphaFold 2 delivered the full 3D structure and made it accessible to everyone.
That database is just Incredible. Hundreds of millions of predicted structures freely available.
It really democratized structural biology overnight. Changed everything.
And the story keeps going. AlphaFold 3.
Just this year, 2024, AlphaFold 3 arrives, and it expands the scope again.
How so?
Now it can model not just single proteins, but complex systems.
Yeah.
Proteins interacting with DNA, RNA, small molecules, ligands, ions.
Okay, wait. So AlphaFold 1, protein shape, basic context. AlphaFold 2, super accurate shape, open access. AlphaFold 3, how proteins interact with other important molecules.
You got it. And it uses a different method, a novel diffusion-based approach.
Diffusion, like spreading out?
Sort of. We'll get into it. But it's reportedly got much better accuracy, especially for those crucial interactions between different biomolecules.
And they have a server now, too.
The AlphaFold server, yeah. Free access for non-commercial research to these new capabilities.
That pace of innovation is just staggering.
It really is, from basic folds to complex molecular machines in just a few years.
I mean, we should quickly recap those versions, just nail down the key points.
Good idea. So AlphaFold 1, 2018, neural nets for contact maps. 1, CASP13, code released, but limited general use.
Okay.
AlphaFold 2, 2020, end-to-end structure prediction, accuracy rivaling experiments at CASP14, open source code, massive open database, game changer.
Right.
AlphaFold 3, 2024, new diffusion approach.
Okay.
Models complex systems, protein, DNA, RNA, ligands, ions, better interaction accuracy, free server for researchers.
Perfect. That lays it out clearly. Progression from context to full structure to interaction. Okay, now let's get into the nuts and bolts. AlphaFold 2. How does it actually work? What's the magic sauce?
Okay, the core idea is leveraging evolutionary information. It starts by finding relatives.
Relatives.
Yeah, similar protein sequences from different organisms. It compiles them into what's called a multiple sequence alignment, or MSA.
Like lining up the family tree of that protein.
Pretty much.
Yeah.
By comparing these sequences, the AI spots patterns. Which bits stayed the same over millions of years conserve residues.
Important bits.
And, crucially, which bits tend to change together. Covariation.
Covariation, meaning if one changes, the other often changes too.
Exactly. Even if they're far apart in the sequence, that's a massive clue. They're probably close together, physically interacting in the 3D structure.
Ah, so evolution leaves breadcrumbs about the structure.
Beautifully put.
Yeah.
Breadcrumbs, yeah. AlphaFold 2 then feeds this MSA, along with the sequence itself, into a very sophisticated deep learning architecture.
Okay, break it down for me.
First main part, the EvoFormer module.
EvoFormer.
Its job is to refine both the MSA information and what's called a pairwise representation, basically. Information about every possible pair of amino acids in the protein.
How does it refine them?
Using attention mechanisms.
Attention. Like the AI paying attention to the most important parts.
Precisely.
Yeah.
Like highlighting keywords. It runs through 48 blocks, iteratively figuring out which parts of the evolutionary data and the sequence relationships are most predictive of the final structure. The MSA info helps understand the relationships, and the relationship info helps interpret the MSA. They feed off each other.
48 blocks. Quite a workout.
It's computationally intensive, yeah.
Yeah.
Then, the refined outputs from the EvoFormer go into the structure module.
And that's where the 3D happens.
That's where it predicts the actual 3D coordinates.
Yeah.
X, Y, Z positions for the atoms, plus the angles, torsion angles that define how the protein backbone and side chains are oriented.
Wow. From evolutionary context straight to atomic coordinates, that's the leap.
It is. And there's another clever trick. Recycling. Yeah, the predicted 3D structure is actually fed back into the network, kind of like an extra clue or template.
Ah, so it refines its own prediction.
Exactly. It loops through this process a few times, improving the structure iteratively.
Okay, and then it tells you how confident it is.
Yes. Crucial part. It outputs confidence scores. PLDDT.
Predicted local distance difference test. Catchy.
Yeah. That tells you the confidence for the local structure around each amino acid. High score means it's pretty sure about that bit.
And the other one? PAE.
Predicted aligned error. That estimates the error in the relative positions of different parts of the protein. So how sure is it that domain A is positioned correctly relative to domain B?
Okay, so PLDDT is local confidence. PAE is global arrangement confidence. Super useful to know where the prediction is solid and where it might be shaky.
Absolutely critical for interpreting the results responsibly.
Right. Now, AlphaFold 3, how does that differ? You mentioned pairformer and diffusion.
Yeah, it's a significant shift in architecture. They swapped the complex EvoFormer for something they call pairformer, conceptually simpler, apparently.
Okay.
But the really big change is the diffusion module. It replaces the structure module from AlphaFold 2.
How does diffusion work here?
Instead of directly predicting coordinates, it starts with noise, a completely random cloud of atomic positions.
Well, seriously?
Yep. And then it iteratively removes the noise step by step, guiding the atoms towards their most likely correct positions based on the input information.
So, like, starting with a super blurry photo and gradually bringing it into focus.
That's a great analogy. It diffuses away the uncertainty to arrive at the final structure. And this approach is inherently better, it seems, at handling multiple molecules interacting like protein with DNA or ligands. It builds the whole complex structure together from that noisy state.
That is a completely different way of thinking about it. Finding the structure by eliminating the impossible from a random start.
Exactly. And that's key to its expanded scope, modeling those vital interactions between different types of biomolecules.
Okay, so with these incredible capabilities, what's the real-world impact been? You said transformative earlier.
Oh, but absolutely transformative. In structural biology, it's night and day.
How so?
Determining structures used to take potentially years, huge amounts of effort, lab work, sometimes luck. Now
you just AlphaFold it. I hear that phrase all the time.
You do. Researchers can get highly accurate predictions, often in hours or days. It's accelerated everything.
Does it replace the experimental methods, though?
Not replace, complement. Definitely complement. The predictions are often used as starting points for experiments.
Ah, like giving X-ray crystallography or cryo-EM a massive head start?
Precisely.
Yeah.
Helping solve tricky parts of the analysis. It's also letting people study huge protein complexes like the human nuclear pore complex.
Yeah.
Things that were just too daunting experimentally before.
And finding unexpected connections.
Yeah, revealing structural similarities between proteins that look totally unrelated based on sequence alone, uncovering hidden evolutionary links.
Okay, what about drug discovery? That seems like an obvious application.
Massive impact there, too. Being able to see the structure of a potential drug target, especially one that was hard to crystallize or study before.
Huge advantage for designing drugs that fit, right?
Exactly. Structure-based drug design.
Right.
AlphaFold, especially AlphaFold 3, with its improved ligand interaction prediction, holds enormous promise here. You mentioned liver cancer earlier.
Yeah, was that an example?
It is. Researchers are using AlphaFold structures to identify new targets and design molecules to bind them, inhibit them, speeding up the pipeline for new medicines, potentially.
Especially for diseases where we didn't know the structure of the key proteins.
Exactly.
And it's not just drugs, is it? Vaccines, enzymes.
Right. Vaccine development. AlphaFold helps design better vaccines, better antibodies, by showing the detailed structure of, say, viral proteins.
For malaria or antivirals.
Yep. It played a role in a malaria vaccine candidate, understanding viral entry mechanisms for COVID antivirals. And enzyme engineering, too, designing new enzymes for industry or medicine.
Making them more stable or more efficient.
Exactly. Tailoring their function based on predicted structural insights.
And just fundamentally understanding disease.
Fundamentally. Getting insights into misfolded proteins in Alzheimer's or Parkinson's. Understanding antibiotic resistance mechanisms at a structural level. Seeing the impact of genetic mutations.
And that database makes it all accessible.
That database is key. Democratizing access. Putting this power into the hands of researchers everywhere, not just specialist labs. It's accelerating biology across the board. Complementing experiments, not replacing them.
It sounds almost too perfect. There have to be limitations, right? Where does it still struggle?
Oh, definitely. Crucial point. It's incredibly powerful, but it's not magic. It's not a perfect crystal ball.
So what are the big ones?
Predicting complexes, especially protein-protein or protein-ligand interactions, where the proteins change shape significantly when they bind. That's still really hard. Even for AlphaFold 3, it's a challenge.
The handshake changes the shape of the hands.
Kind of, yeah. Capturing that flexibility and just the general dynamic nature of proteins.
They don't just sit still, right? They wiggle, adopt multiple shapes.
Exactly. Proteins are dynamic. They breathe, they move. AlphaFold mostly gives you one static snapshot. It struggles with intrinsically disordered regions, parts that don't have a fixed structure.
So it's like taking that photo of a dancer. You get a pose, but not the whole dance.
That's a good way to put it. Predicting the actual folding pathway, how it gets from the sequence to the final structure that's still largely out of reach.
Then subtle changes, like one tiny mutation.
Accurately predicting the effect of single-point mutations on structure, stability, function. Still very challenging. The effects can be subtle but critical.
What about proteins with no known relatives? The orphans?
Yeah, predictions can be less reliable there. Remember, it leans heavily on that evolutionary information from MSAs if there aren't many relatives known.
Less information to work with.
Exactly. And while AlphaFold 3 is improving, accurately factoring in things like ions, cofactors, post-translational modifications, still areas for development.
And there's that philosophical debate too, isn't there? Does it understand physics or just recognize patterns?
That's the ongoing discussion. Is it truly learning biophysical principles, or is it incredibly good at pattern matching based on the vast amount of data it was trained on? Probably a bit of both, but the question remains.
Any other quirks?
Occasionally, the predicted structures might have minor, chemically unrealistic details, stereochemical issues that need a bit of cleanup after prediction.
So it's a fantastic tool, but definitely not the final word. Experimental validation is still key.
Absolutely essential for validation, for dynamics, for interactions, for things AI just can't capture yet. The future is integration. AI and experiment working together.
Okay, so looking ahead then, what's exciting? What's coming next?
Oh, the future's incredibly exciting. We'll definitely see AlphaFold 3 get even better, more accurate interactions, handling bigger, more complex systems.
And tackling dynamics. That seems like the next frontier.
That's a huge area of focus, yeah. Finding ways to predict or simulate how proteins move and change shape. Beyond prediction, though, there's design.
Designing new proteins.
Yeah, AI tools like Rose-Tailfold, Protein MPNN, they're getting amazing at computational protein design. Combine that with generative AI, we could be creating entirely novel proteins for new medicines, new biomaterials.
So not just understanding life's building blocks, but creating new ones. That's mind-blowing.
It really is. And we're seeing new AI methods emerge specifically aimed at dynamics, maybe manipulating those evolutionary signals differently or combining AI with experimental data that actually measures motion, like spectroscopy.
And more integration, AI with other tools, with experiments.
Definitely. Tighter integration, maybe even feeding into automated research platforms, self-driving labs that can design, predict, build, and test hypotheses much faster.
Wow. It really feels like we're on the verge of a new era in biology. Our ability to understand and even engineer life at its most fundamental level is just accelerating like crazy.
That sums it up perfectly. AlphaFold's impact is revolutionary, compressing decades of work, democratizing structures via the database.
The Nobel Prize was definitely well-deserved recognition.
No question.
And yeah, there are limitations still. But the pace of progress with AlphaFold 3, with protein design, with dynamics, it points to a future where our grasp of proteins is just going to reach incredible new levels.
Which leaves a final thought for you listening. Think about this power we're gaining. The ability to accurately predict and maybe soon routinely design these fundamental machines of life. What unforeseen possibilities, what new challenges will that unlock in the years ahead? It's a future that's still unfolding, full of potential.